Mutations or point mutations are which elucidate about the molecular basis of gene mutations. That means DNA is the main genetic material in every cell division process. During the cell cycle, DNA undergo a process of replication. So sometimes shows inaccuracy which may occur within the DNA due to some internal or external or artificial factors. During the replication process, the DNA may not continue its accuracy. The accuracy a little bit is changed in the next generation or next daughter DNA molecules. This process occurs due to some internal factors, sometimes external or artificial factors. The slightest inaccuracy at any level of these levels introduce the changes in the arrangement of nucleotides in a polynucleotide chain of DNA molecule. That means DNA made up of with millions of nucleotides, you know very well. So the sequence of the nucleotide which is altered changes, it leads to different variations. Why we can call as it is a point mutation or gene mutation? Gene is nothing but the tiny or smallest part of the DNA. The smallest change may involve the addition, deletion, or substitution of a single nucleotide pair in the DNA molecule, which may changes the reading frame of the genetic code and ultimately may be manifested into a altered phenotype. అంటే DNA మీద ఉన్న వరుసగా ఉన్నటువంటి కొన్ని వందల న్యూక్లియోటైడ్ల యొక్క వరుస క్రమము అడిషన్ వల్ల అంటే సంకలనము లేదా we can call as deletion లోపం వల్ల లేదా ఆల్టరేషన్ ఆఫ్ ద న్యూక్లియోటైడ్ మార్పిడి వల్ల అంటే నత్రజని క్షారం యొక్క మార్పు వల్ల ఈ జెనెటిక్ కోడ్ యొక్క రీడింగ్ ఫ్రేమ్లో చిన్న మార్పులు చేపట్టబడి అది ఇంకొక రకమైనటువంటి దృశ్యరూప లక్షణాన్ని వచ్చే తరంలో కలగజేయడానికి స్కోప్ ఉంటుంది. కాబట్టి a small minute change in the genetic code frame due to alteration of nucleotide. So if such type of mutations leads to changes, that's why it is called as point mutations. Mutations are include very limited segment of DNA. When compared to the chromosomal mutation, gene mutations are limited to a small portion or a small segment of DNA. That's why it is called as point mutations. I think it's clear. Okay. So point mutation is nothing but a change of a single nucleotide. It includes what? Deletion, insertion, or substitution of one nucleotide in a gene. So these substitution mutations again classified into two types. Substitution mutations are classified into two types. So for example, you can go for, so the substitution mutations are broadly divided into two types. One is transition, another one is transversions. Transitions nothing but replacement of a purine or pyrimidine by another purine or pyrimidine in a polynucleotide chain, that is known as transition. Transversion means the second type is transversion. Transversion is nothing but replacement of purine by a pyrimidine or vice versa. It is called as transversion. So that means when we can call as transitions. So transitions are nothing but the substitution. This is a purine. It is substitute with another purine. So the substitution takes place between the like nitrogen bases. It is PY. PY means what? Pyrimidine. So pyrimidine is replaced with another pyrimidine, we can call as transition. So transition is nothing but in a polynucleotide chain, the nitrogen bases substituted with like base, similar nitrogen base. That means purine is replaced with purine, or pyrimidine is replaced with pyrimidine. That is called as transitions. Okay, transitions. So what about transversions? Transversion is, this is another substitution type of mutations in the genetic material. In this case, purine is replaced with pyrimidine, not purine. So unlike bases, the substitution takes place between unlike bases. The substitution takes place like bases. So purine is replaced with pyrimidine, or pyrimidine is replaced with purine. So such type of point mutations are called as transversions. Is it clear? So the transitions takes place in the genetic material by 4 type of process. One is tautomerization, another one is ionization, third one is base analogs, fourth one is deamination. So 4 type of processes are there under transitions. So one is tautomerization, second one is ionization, third one is base analogs, fourth one is deamination. So transversions also takes place by different alkylating agents and depurination. These 2 processes are relevant to the transversions. So we can go for the slide and we can explain. I think it is not visible clearly. Transitions and transversion. So tautomerization is nothing but what? In the nitrogen bases, the functional group, amino group, imino group, keto group, and aldehyde group. So one of the nitrogen base shows a particular functional group in the cyclic structure. In the amino form, the nature of the nitrogen base different when it turn into the imino form. In the same way, one of the nitrogen base in the keto functional group, the nature of the nitrogen base is differ when it turn into the OH group, alcohol, keto to alcohol. So aldose, ketose, such type of functional group differences are occurs in the molecular structure of the nitrogen base. The nature of the nitrogen base is different. That's why the normal nitrogen base is converted into unusual base, nitrogen base. The unusual nitrogen base, it combine or it bind with different nitrogen base, not like, for example, I will go through AT, adenine and thymine, adenine and thymine. So AT is the one of the complementary nitrogen base pair. So in the AT, so adenine or thymine, in the next replication cycle, it separate, T is bind with again A and it constitute as a normal pair. But the A is when undergo tautomerization, the A star, the unusual base pair adenine is bind with C in the next replication process. That's why in the next generation, always AT pair, For example, I will go through like this. This is A, this is T. So when it is a normal replication process, A again combine with what? T. Yes. In the same way, T again combined with in the next replication A. That means it is a unique complementary nitrogen base. But this adenine undergo tautomerization process. It converted into unusual base pair. What is that? Unusual base pair. This unusual base pair, it combined with a different nitrogen base. Okay. So in the intermediate level, you already studied ATGC. Yes, ATGC. According to the Chargaff law, a purine is always bind with a pyrimidine. So AGR purines, TCR pyrimidines. So A is generally in the normal pair, it is 80. When it is undergo tautomerization means amino form, immunoform it converted into. That is the replacement of proton on the carbon. So this A star, it bind with what? C. So here T is replaced with, so C, and purine is replaced with, due to the tautomerization, it will replace with what? Pyrimidine. So such type of condition we can call as tautomerization. In the same way, in the similar way, we can explain another processes. So that is, see here, tautomerization. Like another one is base analogs and ionization, deamination. Deamination means what? D means remove amino group from the nitrogen base. When amino group is removed from the nitrogen base, it acts different way. It combines with a unusual base pair. In the same way, base analogs. Some chemical compound structure is very close or very similar to the actual nitrogen bases. That is adenine and thiamine, like that. So these, in the place of actual nitrogen base, the base analog is substituted. The base analog bind with another nitrogen base. That's why we are getting AT pair, it become as a GC pair, like that. So that is the base analogs are seen. So base analogs are nothing but some chemical compounds have similar molecular structure to the nitrogen bases present in the DNA. These are called as base analogs. So what are the base analogs we can observe? So for example, the artificial base analogs are 5-bromo-uracil, 5-iodo-uracil, 5-bromo and 5-methyl cytosine. These base analogs show similar structure with the thymine, and the 5-methyl cytosine is related to the cytosine. So when the actual nitrogen base is replaced with base analogs, the base analogs combined with different nitrogen base. So in this way, substitution mutations takes place in the DNA molecule. So this is substitution. So what about the frame shift? Frame shift is nothing but one more base analog is there. Aminopurine is a chemical artificial base analog to the adenine. Aminopurine is also example. So deamination, I'm already told, that is a removal of amine group from the nitrogen base. So when it is removed, it converted into a substitution pair. Transversion. Transversions are nothing but the replacement of between, or the replacement of nitrogen bases, which belongs to unlike, or purine is replaced with pyrimidine, or pyrimidine replaced with purine, like. So these are the transversions, maybe occurs by depurination and also some alkylating agents. So different type of alkylating agents, removal of purine from the DNA strand, that is called as depurination. Due to the loss of the purine in the DNA molecule, it leaves a small gap. At the time of replication, any one of the 4 bases can possibly inserted at this place, and it next level it combined with complementary nitrogen base. So that is about the transversions. So next one is frame shift. Frame shift, it is also, frame shift is nothing but deletion or additions. Sometimes delete the nitrogen bases from the DNA reading frame or codon reading frame. Sometimes addition, a new nucleotide added into the DNA reading frame, codons of the DNA reading frame. So it is also leads to alter the polynucleotide sequence, deletion or insertion of the nitrogen bases, generally which causes by acridin dyes, that is mean 5-amino acridine or proflavin. These are called as intercalating substances. So by this, I will finish the biochemical mutation part. Are you understood?
Yes, sir.
Respond, my dear friends, respond.
Are you followed?
Harish, Dashrath.
Clear, sir. Clear.
Okay. Okay, sir.
Okay. Thank you very much, sir. Mutation is the most important topic for all competitive exams, especially molecular biology, biotechnology, genetics.